Hello, everyone. Today we will be reviewing the kidney's major structures, blood flow through the kidney, and the process of blood filtration and urine creation. The kidneys are bean shaped organs responsible for filtering our blood and extracting waste, while maintaining electrolyte and fluid balance in the bloodstream. Each kidney is located on either side of the spine, posterior to the abdominal aorta. They are located between the T12 through L3 vertebrae, and the upper regions of the kidneys receive additional protection from the 11th and 12th ribs. Each kidney is directly covered in a fibrous capsule composed of a dense, irregular connective tissue. This is surrounded by a layer of protective fat called a renal fat pad. This provides cushion. This layer of fat lies within a strong renal fascia, which works with the surrounding peritoneum to secure the kidneys to the posterior abdominal wall. Sitting on top of each kidney and still covered by the renal fat pad and renal fascia is the adrenal glands. These secrete the hormone aldosterone, which is important for regulation of renal function. The outermost layer of the kidney's internal tissues is the renal cortex. Here we can see that the extensions of the renal cortex project inward to form structures known as renal columns. These serve as structural supports for any important vessels entering the kidney. The renal medulla is located in the inner region of the kidney and is separated by the renal columns into cone-shaped like regions of the medulla known as renal pyramids. Located at the top of a renal pyramid is the renal papillae. This is the site where the complete product of urine is drained into first. The hilum of the kidneys serve as an entry site for the supporting structures such as the renal nerve, renal vein, and the renal artery, as well as the ureter. These are able to enter and exit the kidney through the hilum. The major and minor calyces are structures that extend from the hilum, and together they form the renal pelvis. This is where urine is collected from the renal papillae. The kidneys contain three major blood vessels: the renal artery, segmental arteries, and the renal vein. The renal artery supplies the kidneys with oxygenated blood directly from the aorta. It enters through the hilum, continuing into the renal pelvis, as you can see in the diagram. The renal artery branches into segmental arteries, which continue to branch into smaller vessels repeatedly until the renal cortex is reached. The renal vein is a large vein that drains filtered deoxygenated blood from the kidneys to the inferior vena cava. In summary, the order of blood flow through the kidneys is as follows. Oxygenated blood from the aorta enters the kidneys via the renal artery. This branches into the progressively smaller interlobular, arcuate, and cortical radiate arteries. The afferent arterioles follow. The blood is filtered through the nephrons before exiting through the renal vein into the inferior vena cava. Nephrons are small functional units in the kidney which cleanse the blood, resulting in a liquid filtrate, which is composed of urea, electrolytes, excess water, glucose, and amino acids. As we discussed earlier, the renal artery branches into segmental arteries and continue to branch until they become interlobular arteries. Afferent arterioles are branches of the interlobular arteries. They branch further, forming the glomerulus. This is a sphere-shaped network of capillaries within which blood pressure is significantly increased. Surrounding the glomerulus is the Bowman's capsule. This is formed from the proximal end of the renal tubule. Together, The glomerulus and the Bowman's capsule form the renal corpuscle. Efferent arterioles branch off of the glomerulus and form a network of capillaries around the rest of the nephron tubule up until the distal end. The renal tubule is comprised of three main sections. The proximal convoluted tubule is the section of tubule after the Bowman's capsule. Its lumen contains microvilli, which increase surface area available for maximum solute exchange. The loop of Henle is a U-shaped loop of tubule which functions as the main site for water reabsorption and maintaining a concentration gradient. The distal convoluted tubule is the final segment of the renal tubule. This is where more reabsorption and secretion of ions occurs to fine-tune the composition of the urine. This will eventually connect to the collecting duct. The collecting ducts collect filtrate from many nephrons at a time, and multiple collecting ducts eventually merge to drain the filtrate into the papillae. Within the renal corpuscle, the Bowman's capsule captures filtrate, which is then directed to the proximal convoluted tubule. To achieve this, filtration occurs through a filtration membrane, which consists of three layers. The filtration membrane starts with the fenestrated endothelium of the glomerular capillaries. These have small pores or fenestrae, which prevent passage of blood cells and large proteins, but is permeable to water in small solutes. The basement membrane is a negatively charged layer of connective tissue which serves as a barrier to medium or large proteins. Here we can see a more in-depth diagram of the glomerular capillaries. The outside layer of the filtration membrane is composed of podocytes, specialized cells which wrap around the glomerular capillary and form finger-like projections as seen here. The spaces in between these projections function as filtration slits. These block molecules smaller than around 25 nanometers. Hydrostatic pressure and colloid forces help regulate filtration. Hydrostatic pressure refers to the physical force that blood exerts against the vessel walls. It results in fluid being pushed out of the capillaries into the interstitial space. A high hydrostatic pressure favors filtration. Colloid osmotic pressure refers to the pressure caused by proteins too large to pass through the fenestrated endothelium. Colloid forces result in fluid flowing into the capillary from the interstitial space due to higher solute concentration inside the vessel. This force increases with higher concentrations of proteins like albumin in the bloodstream. When the sum of colloid capillary forces and the hydrostatic pressure of the interstitial fluid exceeds the hydrostatic pressure in the capillaries, this opposes filtration. This is just a more in-depth labeled diagram that I included in case anyone finds it useful while studying. Lastly, we have the countercurrent multiplier system. The nephron loop, vasa recta, and the countercurrent multiplication system works together to concentrate urine and preserve water. The loop of Henle contains a descending and an ascending portion. The membranes of the descending loop contain aquaporin channel proteins. These allow free passage of water into the interstitial fluid. This increases filtrate osmolarity. In contrast, the ascending loop does not have aquaporin channels and thus is impermeable to water. However, its membrane does contain sodium-potassium pumps and leaky tight junctions for sodium and chloride ions to be reabsorbed. Using ATPS energy, sodium ions are actively transported into the interstitial fluid while chloride ions move down their electrochemical gradient through the junctions. This creates a salty environment within the medulla. This salty environment becomes hyperosmotic, which becomes the drive for water reabsorption in the descending loop. These forces work interchangeably to fine-tune the concentration of urine and preserve water. This is the mechanism known as the countercurrent multiplier system. This is another in-depth diagram that I included in case anyone finds it useful. Here are my sources, and thank you for listening.